Hi dear students, welcome to Kavitha's NEET classes. This lecture is the part 3 of the structure of DNA. I have already posted 2 videos on the structure of DNA. In case you haven't watched that, please watch those 2 videos first before you watch this video. So in the previous lectures, I have already discussed the salient features of DNA in depth. So in this lecture, I will just be brushing to the features once and I will be solving the previous year questions which have been asked from this topic, structure of DNA. So if you want a detailed explanation of the salient features of DNA, please watch the part 1 and part 2 of structure of DNA. So what is the salient features of DNA? DNA has 2 polynucleotide chains which are antiparallel to each other and they are having polarity, that is they have having antiparallel polarity, that is one strand runs in the 5' to 3' direction and the other one runs in the 3' to 5' direction. So what we mean by this polarity? Polarity means at the one is 5' and the other one is 3'. What do you mean by antiparallel? Antiparallel means they are running in the opposite directions. So at this 5', why do we call it as 5'? So in the DNA, in the 5th position of the carbon atom, at one end we have the phosphate group. That is why we call it as 5' phosphate, and on the other end at the 3' carbon atom we will have hydroxyl group. That is why we call it as 3' OH. So this is what we mean by polarity. So the 2 chains have antiparallel polarity and the backbone of each of the polynucleotide chain is composed of the pentose sugar and the pentose ribose sugar and the phosphate. So it is the phosphate group and the ribose sugar which forms the backbone of DNA, and from this backbone you can find the nitrogenous bases like adenine, guanine, cytosine and thymine projecting. Okay, so the nitrogen bases are of the 2 polynucleotide chains form complementary pairs. So adenine always base pairs with thymine with 2 hydrogen bonds and guanine always base pairs with cytosine through 3 hydrogen bonds. So a large size purine is always comes to opposite to a small size pyrimidine, and this generates 2 uniform strands of the helix. So here I already told you adenine base pairs with thymine through 2 hydrogen bonds and guanine base pairs with cytosine through 3 hydrogen bonds. And the double chain is coiled in a helical fashion and the coiling is right-handed. So the DNA is coiled in a right-handed fashion, and then this coiling produces major grooves and minor grooves, and the one turn of the DNA is called as the pitch, and pitch of the helix is 3.4 nanometer or 34 angstrom unit with roughly 10 base pairs in each turn. And then the planes of the adjacent base pairs are stacked over one another along with hydrogen bonding which confers stability to the helical structure. Okay, so complete explanation on the structure of DNA is available in the first 2 videos, so please go through that once. So this is the structure of the DNA. What is it that holds the 2 strands of the DNA together and gives it stability? First and foremost, it is the hydrogen bonding between the complementary nitrogenous bases in the 2 chains. So here, see, here there is adenine, here there is thymine, and here there is 2 hydrogen bonds. And here between guanine and cytosine we have 3 hydrogen bonds. So it is the hydrogen bonding between the purines and the pyrimidines which is giving stability to the 2 DNA strands. And then that's the first important thing. Second one, the hydrophobic interactions between the nitrogenous bases also gives the stability, and third one is the base pairing between the purines and pyrimidines. Okay, so it is the hydrogen bonds between the complementary nitrogen bases of the 2 chains and then the hydrophobic interactions between the nitrogenous bases and the base pairing between the purines and pyrimidines which confers stability to the double helical structure. Then again we have sense and antisense RNA. When we study DNA transcription, that is, a double-stranded DNA molecule gets converted to a single-stranded mRNA molecule, isn't it? So, which strand is it actually decoding? DNA is a double-stranded molecule, whereas RNA is a single-stranded molecule. And during the process of transcription, a double-stranded DNA molecule is getting converted or decoded into a single-stranded RNA molecule. So what does that mean? Only a single strand of DNA is getting decoded and the other one is not. So out of the two strands of DNA, only one strand stores the genetic information, and that strand is called as the antisense strand. Okay, out of the two strands of DNA duplex, only one strand stores the genetic information, and that acts as a template for transcription. So it is the 3' to 5' strand which is the template strand for transcription. It is only this strand which contains the genetic information, and this strand is called as the antisense strand. See here, sense and antisense strands of DNA. So we know that DNA is a double-stranded molecule, whereas RNA is a single-stranded molecule. So what happens during transcription? We have a DNA which is a double-stranded molecule getting converted to a single-stranded mRNA molecule, isn't it? A double-stranded DNA molecule is getting converted to a single-stranded molecule during the process of transcription. So, which is the strand which is getting transcribed? So, which is the strand which is containing the genetic information which need to be decoded? So, out of the two strands of DNA, both the strands do not contain the genetic information which is supposed to be transcribed. Only one strand, the strand which runs from 3' to 5' direction. This strand is called as the antisense strand of DNA, and it is this strand alone which contains the genetic information, and during transcription only this strand gets transcribed into mRNA. Whereas we have another strand in the double helical structure which is complementary to this antisense strand, and that strand is called as the sense strand, and it has a 5' to 3' polarity. Okay. So did you understand what's the meaning of sense strand and antisense strand? Antisense strand has a polarity of 3' to 5', and it is this strand which contains the genetic information, and it is this strand which gets transcribed during transcription and gets converted to mRNA. See, in DNA we have two strands, isn't it? One strand runs from the 5' to 3' direction, and the other strand runs from the 3' to 5' direction. So this strand which is running in the 3' to 5' direction, this is the antisense strand, and this is the strand which contains the genetic information which needs to be transcribed. Whereas the other strand, this strand is the sense strand, does not contain any genetic information, and it is not transcribed during transcription. So it's better to know a little bit about the denaturation and renaturation of DNA. So the conversion of a double-stranded DNA to a single strand by heating it is called as denaturation. So here you can see this is a double-stranded DNA molecule. When you apply heat, it unwinds or uncoils and it becomes single-stranded DNA. That process is called as denaturation. And then when the denatured DNA is incubated at a low temperature, that is, you reduce the temperature and you incubate it, then the two strands which have separated, they will recoil. Okay, they will reassociate to form the DNA again, to form the double helical structure again. This process is called as renaturation. So did you understand what do you mean by denaturation and renaturation? The process by which That is, we apply heat to a DNA, the two strands of the helical structure will unwind or uncoil and it will become two separate strands. That is called as denaturation. Once again, if you incubate it at low temperature, they will rejoin, reassociate, and they will form the regular double helical structure. That is called as the denaturation. Now we shall move on to few questions which were asked in the previous year examinations. So these are all the questions on this topic, the structure of DNA, which were asked in the previous year questions. We shall discuss we shall discuss each one of them. If adenine makes 30% of the DNA molecule, what will be the percentage of thymine, guanine and cytosine in it? Okay, so we know there are purines. Which are the purines? Under nucleotides we have purines and we have pyrimidines. Okay, the purines are adenine and guanine and the pyrimidines are cytosine and thymine. And in case of RNA we have another pyrimidine called as uracil. So what are they telling here? They are saying that adenine makes 30% of the DNA. So we know that according to Chargaff rules, the amount of purines and the pyrimidines will always be equal. That is, A plus G will be equal to C plus T. This is one rule of Chargaff rule. The other rule says that the amount of adenine will always be equal to that of thymine and the amount of guanine will always be equal to that of cytosine. So you have to recall the Chargaff's rule to solve this question. So here they say that adenine makes 30%. So here they are saying adenine is 30%. So what will be the percentage of thymine? That will also be 30%. So here you can check in the answer option. So thymine 30%. So this is the right answer option. You need not even solve what is the percentage of G and C. Automatically, just if you know that according to Chargaff rule, the amount of adenine will be equal to the amount of thymine, you can instantly choose this answer option. Got it? Let's move on to the next question. So the answer is D over here. Which of the following statements is correct? Adenine pairs with thymine through two hydrogen bonds. Definitely that is correct. So you need not even go through the other answer option. Straight away you can take the answer as A. We know that adenine always base pairs with thymine. Adenine always base pairs with thymine through two hydrogen bonds and guanine always base pairs with cytosine through three hydrogen bonds. Next question. If the distance between two consecutive base pairs is 0.34 nanometer and the total number of base pairs of a double DNA double helix in a typical mammalian cell is 6.6 into 10 to the power of 9 base pairs, then the length of the DNA is approximately. So how do we calculate the length of the DNA over here? You have to multiply. Okay. So you have to multiply 0.34 into 6.6. You get approximately 2.244 something you get. When you round it up, you get 2.2. So 2.2 is the right answer. Sorry, it is not 2.0. It is 2.2. This is the right answer. Purines found both in DNA and RNA are. What are the purines? Adenine and guanine, which is present both in DNA and RNA. So adenine and guanine is the right answer. The 3' to 5' phosphodiester leakages inside a polynucleotide chain serve to join one nucleotide with another nucleotide. So Phosphodiester bond. We have already discussed what is a glycosidic bond and what is a phosphodiester bond. So it is a phosphodiester bond which links two nucleotides. Therefore, the right answer is one nucleotide with another nucleotide. See, here you can see this is the phosphodiester bond. So this is one nucleotide, isn't it? This is one nucleotide and this is another nucleotide, and it is the phosphodiester bond which is linking them both. So the phosphodiester bond links two nucleotides. Whereas what is a glycosidic bond? The glycosidic bond links the nitrogenous base with the pentose sugar. So this bond here is called as the glycosidic bond, and this bond is called as the phosphodiester bond. So if you know what is a phosphodiester bond and what is a glycosidic bond, very easily you can mark the answer within a minute, within few seconds. In the DNA molecule, the proportion of adenine in relation to thymine varies with the organism? No. See, according to Chargaff's third rule, it states that the amount of adenine, that is AT ratio, is constant for a species. However, it varies from one species to another species. So there is no mention of variation within the organism. So this is wrong. Then there are two strands which run antiparallel, one in 5' to 3' and the other one in 3' to 5'. That is correct. So this is the right answer option. So you need not even go through the other two options because you know that the DNA strands are antiparallel. One runs in 5' to 3' direction and the other one runs in 3' to 5' direction. Which of the following pairs of nitrogenous bases of nucleic acid is wrongly matched with the category mentioned against it? So we know what are the purines. Purines are adenine and guanine, and the pyrimidines are cytosine and thymine, and in case of RNA we have uracil in place of thymine. So here you need to check the right which of the following nitrogen bases are wrongly matched. So which is wrongly matched here? Adenine and thymine is wrongly matched because adenine alone is a purine whereas thymine is a pyrimidine. So that is the right answer. So antiparallel strands of DNA molecule means that, what do you mean when we say antiparallel? That means the phosphate groups at the start of the two DNA are in the opposite position. So D is the right option. This is a factual question again. One turn of the helix in a B-form DNA is approximately 3.4 nanometer or 34 angstrom unit. The following ratio is generally constant in a given species. So here they are speaking about the third rule of Chargaff rule, isn't it? So it is the AT ratio which is constant for a species. AT is here, isn't it? So C is the right answer option. In a DNA, percentage of thymine is 20%. Then what will be the percentage of guanine? So percentage of thymine is 20%. Okay, so we know that adenine and thymine will be equal always, isn't it? So adenine will also be 20%. So what will be the guanine and cytosine? So here 40 is gone, so remaining is 60. So it will be 30 and 30. So C is the right answer option. DNA is mainly found in nucleus only. So now just we shall have a quick revision of the important terms in this topic, the structure of DNA. What is DNA? DNA is the genetic material in all living organisms. In most of the plants, animals, viruses, bacteria, DNA is the genetic material which contains genes which are responsible for transmission of characters or traits from one generation to the next generation. It is called as deoxyribonucleic acid. So the difference between DNA and RNA is this. So in the RNA, the sugar is ribose, whereas in DNA, the sugar is deoxyribose. So in a deoxyribose, in the carbon 2 atom, we have hydrogen atom in the second carbon atom, whereas here we have the hydroxyl in the ribose sugar. So this is the difference between deoxyribose and ribose. Then what is RNA? So in certain organisms, in certain viruses like retroviruses, we have RNA, which is a single-stranded molecule, as the genetic material. Okay, so RNA also contains purines and pyrimidines. So the purines in DNA are adenine and guanine, and the pyrimidines are cytosine and thymine. And in RNA, again the purines are adenine and guanine, and the pyrimidines are cytosine and uracil. So in place of thymine, in RNA we have uracil. Fine. So what are purines? Purines are two ring structures, adenine and guanine. And then pyrimidines are one ring structures, that is cytosine and thymine. Cytosine and thymine. Then base pair. Base pair is a pair of nitrogenous bases, most commonly one purine and one pyrimidine, that are connected through hydrogen bonds in a double-stranded DNA molecule. So what do we mean by base pair? This A double bond T. Okay, adenine base pair, adenine bonding with thymine through two hydrogen bonds is called as one base pair. Here guanine pairing with cytosine through three hydrogen bonds is called as another base pair. So we count the base pairs as 1, 2, 3, 4 and so on. Okay, what do you mean by base ratio? It is the sum total of the adenine and thymine to the total of guanine and cytosine in a DNA sample. That is the ratio of adenine plus thymine is to guanine plus cytosine. This is called as the base ratio. So did you understand what is a base pair and what is the base ratio? This is the base pair and this is the base ratio. See, these important terms and understanding of these will help you solve many questions without memorizing. Okay, so there will be certain questions where you are clueless. However, at least you will be capable of eliminating one or two options, and from the remaining one or two options which you know might contain, which might be the right answer, you can even go for a guess sometimes. So understanding these concepts plays a very important role in answering the in choosing the right answer. Then I already told you what is glycosidic bond and phosphodiester bond, isn't it? So with this, we come to the end of this lecture on the structure of DNA. If you have any queries, you can comment in the comment section below. You can also subscribe to my channel and you can connect with me on my social media handles. Until we meet in the next lecture, take care, bye, have a great day.